Hello. So as you can see, the title of this is Encryption and Steganography of Artificial Gene Circuits. And that might be a mouthful, so I'll make it a little easier to understand. Okay, this might be a little much. If you're wondering, I encrypted the message on the screen. As you can see, talked about encryption in the title. And essentially, encryption is taking a piece of text and putting some key into this sort of function, and you get some sort of like blah out of it. And then to decrypt it, you just take the blah, put a key back in, and then you get the text again. And essentially, this function is used for a lot of different things. It's a very oversimplified, but it's essentially how our passwords work on the internet and whatnot. So if we decrypt it, we get our title back. And I think now we have a better sense of what encryption is, if any of you are wondering. So now let's dive into some background. So this paper kind of focuses on a topic of science known as synthetic biology, which is a growing field that essentially applies engineering to biology. And there are a lot of ways that you can go about this. But in general, synthetic biology is already very important to our lives. It's how we make a lot of synthetic materials for our cars, for plastics, for medicine, agriculture. And you can see that a lot of these molecules on the bottom, they're rather simple molecules that consist of like some carbons, hydrogens, oxygens. And what this paper is essentially anticipating is a growth in this field, where we're going to make more complex molecules from more organisms, and we're going to take advantage more of this idea of synthetic biology. So the purpose of this paper is essentially focusing on a thing called gene circuitry, which is engineering the genes in cells in order to do certain logical operations. And if you're familiar, if any of you have taken comp sci, it's kind of like thinking about logical gates, like and or not. Like if you have two of these genes present, then another, well, no, like two inducers present, then this final thing will happen, and then there's or, not, whatnot. So this paper is anticipating a growth in the value of this and more gene circuits being made. And as such, it is necessary that we should protect the intellectual property contained in these circuits. So that's where this paper comes in. So the two methods of protecting the intellectual property are encryption, which I talked about earlier, and steganography. And steganography, I didn't know it when I was first reading this paper, but essentially hiding data within other data. And you can see that, we do that a lot in disks. And you can see it in this photo, like this image has like a 3, 4, and 5 in it. And like all of those, but when you filter it out, you get all three numbers, can't count, in their own certain colors. So essentially, you're hiding the data when you have it in this form. But when you filter it out, when you fix it in a certain way, you get the data back. So without further ado, let's dive into the encryption part of this paper. So the circuit they were looking at was a pretty simple AND gate. So if LAC-L and ERA-C are both present and these two inducers turn the switch on essentially, then we get GFP produced. And if any of you, everyone's taking biology here, GFP is like that fluorescent protein, green fluorescent protein. And it's essentially, and they're going to use that to like test the viability of these circuits. Any questions so far? All right. So the way they encrypted this, we're using these special little things called unidirectional recombinases. It's a handful, mouthful. But essentially they put these into the original circuit, which we see above, and these two recombinase sites are placed and it flips whatever is in the circuit, well, in the gene. And then they do that with multiple layers, and in between the first and second layer they add this thing called the decoy promoter, which essentially acts as a sort of password. It makes this a much stronger encryption. And as you can see, it strengthens it, and then for decryption you essentially have to go backwards, scramble it the other way, and you have to take out the promoter at the correct point, or else you will get an incorrect scrambling. So A shows a correct decryption, B shows an indirect decryption. So in order to crack this sort of code with like brute force, if you don't know anything about it, the only way to really to fix it is to you have to try essentially n factorial ways, which is quite a lot, and which makes this a little powerful. But that's like the first option, if you construct all circuits based on every gene present. The second option is a little better. It's essentially trying all the possible orders of the recombinases. And I don't actually understand the math, but but essentially you're taking the number of recombinases and the number of recombinases used in possible decryptions, because remember, you don't know how many and where they're using it. But essentially you're summing all of the permutations up. That's the permutation function. But you don't really need to know the math. Essentially, just know that like this is the complexity of cracking it, and it's pretty difficult to do. And now we can go into the second part of the paper, which is a little more complicated, and it looks into steganography. So the way this is done is a little different. Instead of looking at the exact genetic code, it is uncamouflaging the circuit by introducing these a molecular key. So essentially, the camouflage circuit is going to have a lot of this fluff, as I like to call it. So like this is the real circuit, this is the fluff, and it's affecting the circuit in a way that'll make it not functional. And then once you add this molecular key, what you're essentially doing is inhibiting all the parts of the fluff that are interacting with this circuit, and then you get the correct one. And once you take the key out, the circuit will work again. So it's a little more, it's more easily reversible than the other type, but we'll talk about that later. So let's go a little more into how this camouflage and uncamouflaging is done. And the way this is done is using something called a CRISPR key. And if any of you know what CRISPR is, it's used to edit genes. And that's why another reason this is very important is because it's applying CRISPR, and it's pretty interesting. But essentially, as you can see in Figure A, this is your normal AND gate that we saw previously. This is the AND gate with the fluff, and then once you put the key in, it inhibits the fluff, and you have the normal thing, and then you can just recamouflage by taking the key out. And the CRISPR key is guided by these things called gRNAs. The G is for guide. That's why I've been guiding it. And essentially, the number of possible gRNA combinations is the complexity of the circuit. It's how many times you would need to. It's essentially how many different types of permutations you have. So the way they tested the efficiency of these circuits was by essentially showing us a bunch of different combinations of gRNAs that would affect it. And there were a lot, and there were a lot of these graphs, which is very confusing in the beginning to watch, to read. Took a little help. But essentially what this is showing is that when you have the A, F, and G gRNAs that are essentially deactivating some parts of this circuit, then you get the working circuit. And that's shown by this lovely flow cytometry histogram, which I will talk about how it works. But first, you can just see how this one is not working because the peaks don't line up properly. And that makes sense, right? Because E and F are like pretty important parts of this circuit, meanwhile A, F, and G are not. And this is like, and you can see how G is really affecting like the main circuit, but E and F aren't. So it's important that this one does that. And essentially why it's so important that these like peaks line up is because this graph, the X is like the fluorescence and the Y is like the count. So it's the number of cells that like follow in this. And essentially the blue and red are the two different types of inducers, as I mentioned, in the AND gate. So when you have both of them present, you're gonna have the correct circuit. But when they're present at different times and they're not working and it's like out of sync, then the circuit's not gonna work properly. So I know that's kind of complex. Yes, Anish?
How does the fluorescence determine what time the inducer is turned on?
The fluorescence determines whether the circuit's working. Because remember, it's the GFP, which is produced after. So essentially, when they're both present in the same, like, when both inducers are there, they create the same amount of fluorescence in the correct way. And if they're creating the wrong amount of fluorescence, then the circuit's not going to work. So there's like a certain expected qualitative response from this. Okay. And as you can see, this just shows the AFG cancels out these certain parts of the circuit, and makes it functional. So essentially cracking the code is 2 to the n, which is better than the previous one, better than encryption, where n is the total number of genes in the camouflage circuit. And I'm not actually sure where they got the number 2 to the n, because I would think it has something to do more with like permutations. But essentially, this number essentially just shows that it is better than encryption usually. And also, just like an important idea is that like they underestimated in this. The numbers here actually represent how many different possible gRNA recombinases the gRNAs could actually be used to stop these. So if you actually add up all the gRNAs, it's a much bigger number. So it can make it a lot harder to crack these circuits. But yeah, they were just underestimating in this paper. So just some conclusions. We talked about encryption and steganography, where encryption is essentially scrambling the genes at the gene level itself, and it's all or nothing compared to steganography, which can be more thought of as like graded changes. And I like the way they mention in this paper, it's digital versus analog. And essentially what that means is like in like the idea of like digital computer science, there's only 0 and 1, right? There's not going to be like these in-between values. Meanwhile, with steganography, you can have more graded changes, and you can see like that in the graph. Like it's very there. You can have like all these like different types of fluorescence. Meanwhile, with the other one, it's really all or nothing, because you can totally mess up the genes like very easily. And there are some give and takes for each one. With the encryption, it's much harder to go back. Meanwhile, with steganography, you can just cut the key out. But also with steganography, you have to keep the key in like a lot in order for it to function well. And they just said steganography wins pretty much in the paper. But I like them both. I thought it was interesting to discuss them. And there's a third approach, I know. So they mentioned it in the end. They kind of like hit you by surprise. This kind of way is a little more interesting, well, in my opinion. So it's like a synthetic genetic code, where essentially the genes, you can't interpret the genetic code because they change the mapping between the codons and the amino acids by putting in these artificial tRNAs. And the reason they didn't actually do any experiments in the paper was because it's still like in the making, this process, and it's pretty complicated. But some proof of principles have been done, and yeah, it's very very challenging to pull off. And also, people may only have interest in the functional cell itself and not the actual code. It means you could just replicate that cell and have it, which is a sort of problem with this, or it's a different strategy, I would say. So just some future research in this sort of way. I mean, just getting back to it, this whole idea is like synthetic biology, right? It's putting computer science and biology into the same sort of field, which has a lot of potential. And yeah, synthetic biology, exclamation point. Yeah, it applies CRISPR-Cas9, which I'm sure a lot of us have heard of, but in like recent years. But you've seen like there's been like a lull in like the sort of like talking about it, but you know it's making a resurgence. And also there's the idea called like plug-and-play cells, which essentially could be very useful for treatments for specifically like cancers. Like if you can put in a cell that can create certain toxins when given like the correct amount of like the correct environment, then you can fight cancers pretty well. There's a lot of potential with this field, and this paper went into like the encryption and not the actual usages of that. And yes, that is all. Anybody have any questions? Okay. I'll have to drop them.